some of you may have heard this, and it's not that different because this will go on and on for next 30, 40, 50 years, even if I'm not there. So probably this will be an issue for many people who are listening to this lecture. So there is a first issue. I think climate change is a big issue. It changes almost everything. See, Korea, we have like a ocean around, and all these fishes are changed because its temperature is going up and up. And we used to see the fish before; they are not there. They are all the way up to the north, and then we have new kind, maybe something close to what you have had 10 years ago. Then now we have it. So things are changing. We have to think about it. See, once it was very cold place now is sometimes very hot. And you can easily find everywhere. So far we have gradually increased temperature elsewhere. It's been in the news and nature editorials and science saying that now we have the planet is heat up for past 30, 40, 50 years. And it used to be the plant doing well in the desert, so they don't have problem even if there's not much rain. Now they have problem with the photosynthesis. The red indicate that they are in trouble with this photosynthesis, but they are not doing well. So that could also change a lot of our environment as well. And this was Greece, I believe, and they had a problem with this in a couple of years ago. Yeah, flooding in central Greece, and they called a disaster. Indonesia, now even bigger problem that since few years back that we talk and discuss about Jakarta is thinking, therefore we have to move this capital to other place, and then all of a sudden that talk stop, I believe, and then the government changes and back and forth, and now we don't know what to do with this. But this is likely within 50, 60 years that many cities are disappearing. That's one of the problems, right? So they back there last year that they said that they will put $35 billion to set a new city, but we have to wait to see. And it's just Iceland that has a volcano. At the same time, there's a flooding in Kentucky at the same day. So there's a lot going on. And also climate change. Once you have it, then there's another problem, outbreak, viral, bacterial disease outbreak. That's the problem. Okay? So the climate change is the problem, not only itself, but it induces other problems that causes domestic animals and humans. I'll talk a little bit more about that later, but say New Zealand, they declare national emergency because of this cyclone hit the North Island. And then there's a power outage reported in all of the countries, and therefore they had a problem for a few days or so. And also, other than those climate change, we have problem with greenhouse problem, and greenhouse gas concentrations are at their highest level in lifetime of the Earth. So we know that greenhouse effect is that we have the sun's radiation travels toward the Earth is coming in, and then they are many of those, about half will be reflected and observed to by cloud to the atmosphere, but that's okay. And also that the rest, they will reach the earth. So when there's a radiation, it comes to the earth and half of them will reflect, and it's not 100% reaching to the earth, but half of them will reach in here. Then they release it back, and then also they pass through the atmosphere. Most of it captures and retained by greenhouse gas. Now, what we say greenhouse gas problem is this one, okay? Number 6. Now we have to think about what to do with this as well. Gene editing issues. To me, gene editing issue, they do have a pros and cons. How wisely we use, and then probably we will get a lot of benefit. If you see the news by CNN and some Nature editorials, and now the United States, they will allow gene-edited pig for human consume. So things are also changed. This has something to do with welfare issue as well. So junior scientists should think about which area to go and what to do and how to do. That will be an issue. So I do not give you an answer, but hopefully I provide some information to you so that you can go over with these issues so that you pick something that will be hot and important and useful and valuable to society and country and among ourselves as a scientist. Okay. So there's another one like a corn, for example, when they use the gene edited one, and it is continuously they report that they yield the better productivity. And soil bacteria could provide third source of nitrogen. So there's another source of nitrogen, which is good for all the soils and plant and eventually to the animal. And as I just said, US has approved CRISPR-Cas9 gene-edited pigs for the food. It's been released just a month ago. So now it will have an impact to other countries as well. For example, South America, Brazil, they produce huge number of pigs. Not much in Indonesia, only two state or province, but still, once they issue like this, then that will have an impact to not only pig, perhaps sooner or later will be cattle as well. Okay, buffalo, maybe, you know. I think buffalo was the topic yesterday or the next one. But and then we know the fishy and salmons and all this. This is ordinary one, regular one. And the backside, this is the gene edited one, at least 3 times bigger than normal one. So you can tell that what benefit we can get to have this. Now, whether this will be welfare issues, maybe not. It is about whether you can produce and you can import or export. Okay, so this is a reality. They already have it. So this is the real alternative meat issue. I think this is the big time. Within next 20 years or so, then we will have huge issues with this alternative meat issue. World population to reach the close to 10 billion by 2050. And then alternative meat to make close to 50 or over 50% the market in 2040. Well, believe it or not, or if it's not the 60%, but still quite increasing these days. This young scientist or a scientist to be in this lecture room, probably this you think that this is not in Indonesia or far away from the Asian countries. That's not true. It's coming. So we have to think about what to do with all these issues. And that has coined with carbon net zero 2050. So see rapid increase of meat demand, increase of population that induced the increase of the meat demand. And now we have issues with food security, resource, animal welfare, food ethics, together with environmental sustainability that I just talked in the previous slide. Now we have to look for eco-friendly, ethical, sustainable source of the meat, plant-based meat, analogous, edible insect meat, and cultured meat. This edible insect is not only applying to human being, but also to the animal as well, domestic animal. Actually, it's a quite hot issue in domestic animal area that how we can use this insect source as a protein. So alternatives to overcome inevitable limitations of traditional animal production. That's quite an important issue. Oh, before I forget, if you have any question, then during the talk, you can ask a question on the chat, please. So there are some choices like this, okay? And infectious disease, unfortunately, that's my favorite topic, and we shouldn't have it, but we cannot get away from this infection. FMDV and avian influenza is a hot issues during past 4-5 years. Why is the problem? Because once there is problem, we have to kill the enemy. Once we do that, there is a problem with economic loss, and then reduce the genetic diversity. And then, of course, there's the producers, and in the field, there's emotional concerns, but also ethical concerns, because we have to kill so many animals at once while they are actually alive yet. And there's a problem with the long-term sustainability once we're stepping out of animal. So this was the editorial that I wrote to Animal Bioscience last year. And there's an issue with H5N1, that's high pathogenic avian influenza. And if you see the textbook in the vet ecology, it says avian influenza never get into the ruminant. But now it's changing. It's changing the whole game. Now, it was found in dairy cattle. It was found in milk. Luckily, that did not spread to the human. But now it's seemingly that we do have cross-species infection. If you think about if that comes to, actually there's one case, or at least one case, reported that farmer get this infection from the animal, but it's not widely spread yet. So we have to watch out closely with this problem. It has been in the United States, now it's quiet, so, and therefore I would say that it's not spread anymore. But you can see many states in here, 13 at least by then, had a problem with this issue. And of course, African swine fever, ever since we have had this year 2023, start from the China, and now it's all over the Asian countries, and now it's a big problem. And not only Asian region, but also EU, they do have a problem as well. So it's elsewhere, we do have African swine fever problem. When we have this disease problem, what's more, there's a market recession. So once we have these problems, COVID-19, we have experienced already, not only the lockdown, but see, there's a problem with animal management. Price of feed goes up, availability of facility, there's a limitation, and there's a limitation to have this feed ingredient from place to place, and transportation and marketing problems, and a service problem, okay? And therefore, there's a shortage of laborers and shortage of food, and everything has to be shrinked. And therefore, there's problem with animal production. And then consumer changes in psychology as well, and change in behavior. And people try to have this huge storage at home to save themselves, but that's still not enough. There's a problem with the security, low economic support, and also there's a danger in food safety as well. Another big time, it is coming to Asian countries from EU, and hit the Asian countries, hit the North America and South America, is animal welfare issues. It started from the EU, and there's a big issue in North America and also EU. It has an issue in Korea. So what we have at the moment, we have shift from regular egg to cage-free eggs. We used to keep this chicken in the wire cage, battery cage. Now people pay more to the eggs that chickens were raised from cage-free. and also animal-friendly products are considered healthier. Well, if we have a nutritionist in here, you may say, no, me, I had my master in nutrition. Well, egg is egg, unless it's rotten. So when it's a fresh egg, then there's not much differences on nutritional value, but people like it, and therefore they pay more. So it's changing. Customer will change the way we think about our product. That's true. So you should think about not what you think, what future customer will think about our product, meat and milk and egg, all those things, okay? So I must say that we do have various psychological and physical stressful conditions on animal farming, and number of different issues that I have raised up to now. I would like to say briefly with a few slides about stress and productivity, why we should care about the stress, why the immune response is important. At the end of the day, everything is with this domestic animal is about enhancing productivity, how you could increase this productivity from the animal of our interest or your interest. The definition of stress, and stress is a response to a stressor. So stressor is root of stress. It could be an agent, it could be condition, it could be environment. So it could be anything and everything that would have an impact to the animals, not only animals, human beings as well. So believe it or not, there is a negative stress or positive stress, both sides, pros and cons. And they have an impact on mental and physical well-being for human as well as the domestic animals. But I'll focus on domestic animals in here. There will be nutritional factors and environmental factors and biological factors and also physical factors. For example, when there is a unbalanced nutrient or a toxin, starvation, diet change, especially for nursery pigs after the or during the weaning period, we know they have trouble and they have diarrhea and they have a lot of problems because this diet is changing from the milk to solid feed. And environmental factors, the temperature, microbiota, past 5 or 10 years that there's a huge issue on microbiota. People who are doing this cattle, especially the ruminant, we know microbiota play essential role to change this diet to short chain fatty acid for energy source. Without that, cattle ruminant cannot survive, that we know from old days. But now this concept is everywhere. Also, biological factors are weaning, infection, and vaccination. Excuse me. And physical factors, transportation, lack of spacious area. All domestic animals, especially the pig, they do have social status and therefore they do have stress conditions. That I call stressor, that all these, each and every one of them will be stressor. In normal condition, if you look at the gastrointestinal tract, and they have a microbiota, as I said, and also they have this first lining of the gut, and they have this mucous layer, and they have tight junction proteins underneath of this intestinal lining that they have immune cells. And if you think about what immune cells are doing there, and when they produce proteins, would that be beneficial to the animal? In fact, that's not exactly true. because they use energy and protein and all this source to make proteins not for productivity. But when it comes to infection, oh, can you hear me okay? I think there's some broken. Is it right? Is that okay,
No,
Dila?
yeah, it's okay.
Oh, okay,
Can hear
good.
you.
Thank you. So but once you have problem with this stressor, then now this changes. What happens? It's seemingly the same, but they have different group of microbiota we call dysbiosis, and there will be a immune reaction. Now they will have impact on productivity. Obviously, when you have this problem, and they will have low productivity. This is the reason why we care a lot about gastrointestinal physiology first, and then immunology. So for better productivity, stressor-mediated microbial change and immune system should be carefully examined. That's a fact. That's a fact. I'm saying based on fact and strong hypothesis. So in our lab of animal immunology, we study immunology, of course, but at least more than half of topics that we work on domestic animals. And the other half, we work on mouse and human as well. And then we work on infection and vaccination. If I have to dissect a bit, then we work on a comparative immunology because of different species, microbes, metabolite, immune modulation, nutritional immunology, and then mucosal immunity, infection and management. But what I emphasize in my lab is we should working on function and mechanism. We are not the producers. There are other group of people, they do produce, but for us, we are the scientists. We should do science. So during the, I don't know, 10, 15 years that we've been working on pig, chicken, and also cattle, now we will have a new cattle scientist coming after Myunggi Dila, and I'll introduce him to you later, not today. So
Thank you,
from
Prof.
next semester, the new professor will come to this ruminant. Actually, he's the student of Professor Ha, Jong-gyu Ha. So, yeah, we'll see each other sometime sooner than later. Wait a while. So we still work on mouse and human as well, and nutritional immunology, as I said, and immune cell regulation, and then mucosal immune modulation, and nanoparticle and immune modulation as well. Somehow. Oh, excuse me. Okay. So from now on, I think I have to brief, and I may have to finish within 10 minutes, and then take some questions and give you a least quick break before you start the next lecture. Sorry. So what we do, we work on immunosecurity. That's what I call. You don't have to call that, but the immunosecurity to me is quite important. So basic research, this has been published. I'll go very quickly. Bursa, that's a very unique organ. Only a bird has. We don't have the mammals. We don't have it. And then looking at all these different B cells and immune cells, how they change. So aim was to looking at dissect and classification of B cells during the developmental period before the hatching, but also after the hatching. So we look change of the cells from embryonic stage 14 days to up until hatching and then thereafter. And we found they do have changes on markers and cells, size of the cells, and then many other parameters before they become peripheral B cells. And that has been published. And we're also looking at another part of immune cell, macrophages. And steady state, they are working on clear the apoptotic cells or the senescent cells. But once there's an infection, it's different. They act as a phagocytosis and then try to clear the pathogen. That we've been looking at in this study, checking all these parameters. So we try to characterize the monocyte and macrophage like cells during the steady state and then during the LPS challenge. We looked at all different organs, what are the differences based on these markers, MRC and MHC2, and we found some differences. And also we looked at what are the differences between those two populations, and we found this uptake is different, much better with this MRC high and MHC low cells. And also we looked at the markers, looking at the mRNA, CCR5, 2, and CXCR1. That also changes. And in order to prove that chemokine receptor migrate toward the this chemokine, so we add chemokine at the bottom and see the migration. We found that this one, CCR5, worked on XCR that we found in the previous slide. So we had proved that that's true. And also we inject LPS to see how those cells are changing. And as predicted, there has been increase in a certain population in here and MRC1 high cells. The region number changed. The reason I'm going really fast is one hand is published. So you can find it if you want to. On the other hand, I don't want to spend too much time what we have done because this is a part of what you should learn. The first half would be much more important, and probably your question is more important than what we have done. I'll show you some of our unpublished data later. So we found this. There are some differences between two different immune cell types in terms of phagocytosis and migration capacity, and also inflammatory condition. The proportion changes, and so as pro-inflammatory cytokines. And this has been also published in vet research. And then we try to see a salmonella clearance by macrophages. And there's not many papers actually. And also they have not used macrophage cell lines or primary cells, although they are quite doing well with this area of the research. So we decided to looking at phagocytosis and opsonal phagocytosis. Difference between these two is that phagocytosis, they do not use any of the receptor, but opsonal phagocytosis, they use this receptor. So receptor mediated, non-receptor mediated. Okay, so I'll go a little fast in here. We found phagocytosis, there is some change with the knockout cell line. Oh, this has been made by Professor Tessa Park that many of you know. So he's the CRISPR guy. So I asked him nicely to make this knockout cell line, and he make it. And we have it, and we have done some studies with Professor Tessa Park. Actually, I'm here at Pyeongchang campus. I'm just next room from his room right now. He's sitting the other room. Without his help, I wouldn't be here connected because I cannot find the password today. But we're looking at the opsonal phagocytosis, and then there's some changes. And it was good that we have Professor Tessa Park, that he made this knockout system so that we can use it and we can find some quite nice significant differences on opsonal phagocytosis. And then we're looking at some transcranial changes. And also we found there's a quite distinguishable upregulation and downregulation of this trace mRNAs. And opsonal phagocytosis is the same. So we're looking at all these toll-like receptor 4 knockout cells behave quite different from this wild type. So I strongly encourage many of you if you do have a chance to meet Professor Tessa Park, then ask him to provide some of these knockouts or to make this knockout so that you can try some of these studies yourself. So we've seen some changes with that. As I promised, I will take just a few minutes and then finish up and then probably take questions from you if you have any. And when these days, past 10 years, we have really working on metabolite. So we're looking at the cell metabolism to see all this analysis and this compensatory glycolysis, looking at mito-OCR and glyco-per ratio. And then you can tell that how it is changed from here. So blocking and then in here. So that's reversed. And so that you can tell that whether there is a reprogramming during the opsonal phagocytosis. All right. So this has done, he just got the PhD, Ki-Sun Yu, or Dr. Ki-Sun Yu by now. And also, we have done some studies. This is also published, and there is a change. If you remove all this microbiota, and there's a changes on the immune cells, so as some proteins produced by those immune cells. And then you put wild type together, and then the microbiota will come back, and then that so as immune cells. And then later we found that that was gram-positive bacteria. And then acetate was the responsible for those changes. Clostridia was the one that had responsible for that. And then this was published already. And I talked about dysbiosis. And you probably know that fluorophenical is still used in the field. And when we use this, we were able to induce a dysbiosis in the chicken. And then we'll try to looking at the in details. This has been already published in Journal of Animal Science and Biotechnology last month or a couple of months ago. So other than those domestic animal studies, we also have done some mouse and human studies as well on B cells and CD4 T cells and CD8 T cells. And now they are at Harvard Medical School for they're doing a postdoctoral fellowship. And he also finished his PhD and then a postdoctoral fellow. And they also finished their PhD, but they working on nutritional immunology in terms of dietary intervention in mouse models and asthma in human. So conclusion is that what we need, I think balance is the key. Nutrition, host immune response, and animal productivity. We have to take all these parameters together. We cannot just looking at diet, feed ingredient, and productivity. We have to take more than that into account. So, and then I emphasize the importance of stress and also immune response has to be balanced well, activation and tolerance. When we work and we had to think about the farm and field and government, university research unit, because the farm follow the regulation, sanitation, vaccination, welfare, those are the issues. But government, they have to set the proper policy and they should have long-term plan and they should have monitoring and support to whom? To field and university and research unit. For us, as a scientist, we should do a science. And therefore, company can make a product to provide to the farm and field nationally and internationally together. I think that's the message. And I would like to give you another one, important one. Soon there will be Animal Bioscience Forum. It's a free. Prepare your phone. I have a QR code for this one. This is free, online, in Korea. I'll be facilitator, but we have speakers from Belgium, United States, Korea, Netherlands, Japan, and everywhere. So take this one. Otherwise, I'll pass it to Dilla later so that she can contribute to the class so that you can come to this class as well. It is about precision livestock farming. transforming the landscape of animal science and industry. So it will be very interesting talk. I guarantee. You don't have to trust me, but I'm sure, oh boy, there are a large number of students from our lab and also international collaborators. And I thank to all these people and also funding body, NRF, RDA, and KHIDI. And thank you very much for your attention, and especially all these listeners and attenders of this lecture. And I grateful for that. Thank you. And Oprah Winfrey says real integrity is doing the right thing, knowing that nobody going to know whether you did it or not. So I'll stop here, and you can send me a question to this email, but also on chat. So I will check the chat, I guess.
Okay. Can we do Q&A, Prof?
Yes, we should have Q&A.
Okay. Thank you, Prof, for the interesting lecture about nutrition for enhancing the immunity and productivity in animal. Now, I will open the discussion. Already one question?
Zaidan?
Yes, Zaidan. Zaidan, please.
Jaju Akila from
Yes.
Indonesia.
Yes, from
You
Indonesia.
can ask a question.
Hello, Professor. My name is Zaidan, and I'm undergraduate student majoring in biotechnology and food nutrition, National Taiwan University. So I also listed my question in the chat box if my spoken question was unclear. So I have three questions in here. So basically, my first question is like regarding the data on the TLR4 knockout macrophage, we saw enhanced opsonal phagocytic activity. Could this be a result of compensatory upregulation of other pattern recognition receptors such as TECTIN1 or MARCO? And if so, how might this alternative ERS contribute to maintaining pathogen clearance in the absence of TLR4? The second one is you highlighted the role of gut microbiota in regulating host immunity response. This made me wonder, is microbiota composition heritable and could it be used as a selection criterion in breeding programs to enhance immunocompetence in livestock? And the last one, you discuss various types of stressor that impair immune function and productivity. In this context, I'm curious, like, what are the epigenetic consequence of chronic stress on immune cell population in animals? And are those changes potentially reversible through not nutritional or microbial intervention? Thank you, Professor. That's all my question.
During the question, I lost you, so I could not listen to you properly.
Oh,
But if you type on the chat, I'll answer the last question because you asked about the stressor and immune cell, how is changing upon the stressor in domestic animals. So I heard the last question, the first two I lost and then I reconnected just now. And therefore, if you, while
oh,
I answer to your third question, and first and second briefly ask that, okay? So third question was whether how the immune cells are changing under the stress in domestic animals. So what I was thinking is that probably depends on how it is changing. It is different. Oh, he typed already in here. Okay, so different situation will change the immune cells differently in each and every domestic animal. So what kind of stressor and what kind of changes and infection will determine what immune cells to be activated in which organ. So you should looking at different immune organs and how it's changed. The first question was, could the enhanced opsonophagocytic activity in TOL4 macrophages be compensated by upregulation of, oh, Jaidan is very good. I didn't expect this kind of question from like, oh
It's
man,
interesting question, Prof.
you should come to Seoul National University to do this experiment by yourself. Very good question. Yes, so this question is not only about the Tolla-Crystal-4 knockout question, CRISPR-Cas9, overall, when you knock out certain gene, there will be some compensation. And for us, we check some of these genes in relation to toll-like receptor 4 gene, and then there wasn't much changes. We have done 2, 4, 3, 6, and double knockout and triple knockout. But this question is really good. We still try to see whether there is any changes because changes of other receptor will have a great impact. This is a very good question. Jaidan, if I come to Bogle University, you should see me, okay? Or is he in the Bogle University?
No, Prof.
Okay, good. Second one, is microbiota composition helpful and usable in breeding programs for, oh, this one is also a good question. We believe we could, but I think our result on our hand based on research done so far is not enough. I think most people are saying that probiotics, but to me, we should have a lot more data on prebiotics and probiotics and postbiotics. I do have interest in postbiotics more than pre and probiotics, or sometimes postbiotics together with bacteriophage. So there are some choices. This one also, probably we should make another small group meeting or chat later, not in relation to our lecture series, so that I can answer to Jaidan better. And Dila, you should arrange our meeting somewhere, or I should
It's
come to
alright.
Indonesia during the summertime. But all these questions are very good. I think if you would like to work on this area, probably talk to Dila or me or Professor Tessa Park. Currently, my professor in National Taiwan University about maybe similar topic. So that's why I want to ask you. Oh, sure. I also come to Taiwan or National Taiwan University. We have some MOU, I guess. And Osra Joel from Uganda, very nice meeting you. Osra, I'm sorry if I misspelled or mispronounced. What property or properties of gene-edited animals make the undesirable for consumption? Oh, okay. It's a good question. I'm sure there are something that would undesirable, but we try not to have undesirable animal production. Mostly we would like to have desirable production. For example, there's a cover story of science saying that cattle has been produced without horn, so that the better productivity. Undesirable, I have to think about it, but we, as a scientist and producers, we try to get away from undesirable for the consumption. So I think I'll stop here for next class, but if you do have a question, then get back to me. And also I have to move on to next lecture to some other country, believe it or not, it's true. And also, Jaidan, you should send me an email so that we can talk
Yes.
more.
Yeah. I think that's
Okay. Thank you, Dila.
Yeah,
And thank you very much, everybody. And I really enjoyed your participation and but also questions as well.
yes.
All right.
All right.
Good. Thank
Thank you,
you.
Prof, for the answer and also the insightful materials.
Sure.
On behalf of the committee, I would like to express sincere gratitude for your presentation today. I hope we can meet again offline someday.
Oh yeah, sooner. Yeah.
Okay, soon, Prof. Okay, thank you very much, Prof. Choryun from Seoul National
Okay.
University.
Thank you.
Yeah, thank you, Prof. Okay, and now we will move to the next lecture. We have Professor Erika Budiarti Lakoni from IPB University. Maybe I will read her CV. Please wait.
Hello, Dr. Dila. Yeah, sorry. I asked what time my lecture is 2 o'clock.
Okay, yeah,
Okay,
you're
yeah.
like 2 o'clock, but I will introduce you first. Please wait.
Okay, thank you.
Okay, Prof. Erika Budiarti Lakoni, she got her PhD in Feed Science and Technology at IPB University, and she is a professor also in Department of Animal Nutrition and Feed Technology, IPB University. Her research interest is about animal nutrition as sustainable feed resource, also about plant-based feed additives, functional feed and cell technology, antibacterial and psychochemical properties of biopolymers, and also feed microbiome interaction. Today, she will deliver lecture about innovative approach to support animal feed and promote sustainable farming system. Are you ready, Prof?
Yep.
Okay, time is your Prof.
Okay, thank you, Dr. Dila. I saw many my students here. This is our Secretary of Departments. So nice to meet you all. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon all. So Dila, that's I think that's my name is correct to the Erika, not by C, by
Yeah,
K.
sorry, Prof.
Okay, yeah. According that's your title for me, maybe I cannot read again my my CV because Ibu Dila already explained more about my CV. Maybe it's wait a bit. I will share. Is it I will share onto my PPT, Ibu Dila?
Yes.
Yes, of course. So yeah. I share it. Oh, okay. Dilihat itu. I think this is my CV. Yeah. Okay. Can you see my CV? This is a lot. This is the true is my name. Erika, not C. Yeah, I already 64 now. So lecture in Division of Feed Science and Technology, Department of Nutrition and Technology. Now, the IPB give me the responsibility, the head of Institute of the Science and Technology part. IPB have the Science Techno Park is one of the Science Techno Park in the university in Indonesia. And before the head of the Institute of Science Techno Park, that's my duty is Vice Rector for Innovation and Business. So I think is it suitable, Ibu Dila give me the title, how the innovation is very important for us. So there's a lot of the member of the Board Trustee and member of the Senate IPB University and member of the professor and maybe the head of the Badan Standar Nasional Indonesia. Now I have the responsibility as the head of the Science Techno Park, but it's dominant for me is a lecture. Whatever my duty in the IPB or the national, the lecture is I think it's very important for me. So I will share my title. Wait. Thank you, Ibu Mbak Dara. I saw my, wait. I stop here. Mbak Dara, are you there? Yeah, thank you, Dara. Mbak Dara prepare my PPT.
Yes, you are welcome, Bunda.
Thank you. Yeah. Is it correct? This is my title. Budila, is it right? The innovation approach to support animal feed and promote sustainable farming system. Is it right?
Right.
Right. Okay. Yeah. And you can see this is the Science Techno Park. IPB have the Science Techno Park. Why we have the Science Techno Park? The IPB said this innovation is very important. If the student have the research, we still call it is the invention, not yet to innovation. So innovation very important to us how to deliver knowledge, our analysis of knowledge and policy become to invention, the result of research, and then become to scale up the innovation and business. So IPB have the Science Technopark. What kind of Science Technopark? Science Technopark is integrated area that it means professionally or innovation development. So I think it's innovative is very important. Business incubation and technology transfer in order to encourage sustainability, economic growth. For the farm system, I think the encourage sustainability economy for animal husbandry is very important. If we still have the animal protein is very important for us. So this is the STP and IPB. Maybe for Ibu Sari, if you have the quotation for visit the STP in Taman Kencana, please send me later. If you want to see because we have the STP. I think from the other department already sent a letter for me to visit the excursion for the summer course. I invite for our department to visit the Science Technopark. Is it possible? Yeah, Udila, is it possible? Yeah.
Yes.
Okay. Yeah. Okay. This is the part Science Technopark in IPB University. We have the teaching industry and we have the big industry and we have the halal incubation and we have the startup building in here. Yeah, in IPB, thinking about this very important for us, innovapreneur, value creation and community industry. So I think it's for our animal husbandry, not only for research, for our knowledge, for scientific, but we have to concern the result of the research go to community and go to the farmer with the collaboration with the community and with the industry. So this is for IPB. IPB have the type. Okay, the IPB have the type entrepreneurial university. Teaching is maybe for in the class we have the teaching, but we have to learn, give to our student entrepreneurship. This is the basic entrepreneurial university is in campus. But after the research we have the invention, and invention give the innovation. Because after we protect the invention, this is the innovative. So the innovative research is very important to us. And then the entrepreneurial. So the IPB have the one basic entrepreneurial university, and the second technopreneurial university, and third is socialpreneur university. And we can combine in here the IPB, where we have the techno-social entrepreneurial university. I think the animal production, not only in the industry, because the animal production still in the community, still in the farmers. So I think this is the techno-social entrepreneurial university in IPB. So this is the techno-social entrepreneurial university, what kind of design technopreneur in IPB. After we had the research, I think it's all the lecture, all the student, and all the staff in our campus collaboration to make the research. After research, we can have the innovation. In Science Technopark IPB, we develop the research and protect the IP, and then make the transfer technology transfer, and then protect the IP, and then we have to scale up. A lot of the funding from the government to scale up the innovation there. So we concern the academic is number one, innovation, and business, the market, financial, and the government, the quality, regulation, and community. This is the animal husbandry in here, farmer mentoring. For design, for the design sociopreneur IPB, this is because I think the animal husbandry in the society more in India. Community learning center have the technology facility and go to the market. We in IPB we already done about this techno-social entrepreneurial university. And what kind the IPB have to promote and to prepare the student? In IPB have the talenta development maybe in the level 1, yeah, Ibu Sari, yeah. Ibu Sari, is this there? Yeah, we have the level 1. We already have the level 1. We call the PPKU, ya, Lila, ya. PPKU,
Yes,
we call the
PPKU.
TPB. We have the Talenta Development. The IPB prepare for our alumni. This is not only just the government employee, but have to business. So we have to go to the business with the innovative, creative, and the other. We have the Talenta Development. So the IPB have mapping organization, have to mapping student potential for the management of the learning information about that. So clustering full talenta in IPB, 40% is professional. I don't know in the other university, but in IPB, now in IPB have the 41% professional. This is the talenta. And the lecture, Budila, is it a worry for us because the only 6%. 6% become to lecture and researcher. So I think we have to think about that. If we got the retired Budila, Busari, and the others, so this only 6%. Stop our alumni and our students want to be lecturing here. And then entrepreneurial. So you look at this data. 43% is become the entrepreneurial. A lot of our students, a lot of our alumni become to the 40%, maybe more the 43%, like the entrepreneurial. So the IPB give the organization the structure, structure it to manage and prepare for our alumni 20 years. Based on creativity and based on innovation. This is the Talenta and entrepreneurship development and the Science Technopark and the other directorate give the facility for our alumni here. So this is the invention downstream, the innovation commercialization. What kind of innovation for the animal husbandry or faculty of animal science in here? In our department, in our faculty, this is the innovation development here. So innovative invention is very important in here because because we need for feed, we need for production and the other, we need the result of research and suitable for our animal here. So that our research not only for publication, for our ranking. So we have to protect IP, intellectual property. Intellectual property is very important for how to make the agriculture or farming system in the field because and in the world now protect the IP because the IP is the result how to protect the invention, how to protect our knowledge in the IP. So this is the community empowerment. So this is the scientific. So what we do in the Science Technopark. In Science Technopark IPB University, we have the training innovation gallery, technical assistant. That's why I invite the members of the summer course, Budila, to visit in our Science Technopark. You can see the gallery innovation, a lot of the gallery innovation here. And IP innovation management and business incubation and industrialization here. So this is all the commercialization is courtesy for the IPB. So a lot of our research is we call the Palais of the Palais of Death. Maybe not only in Indonesia, a lot of our research it's not have the many function for our society because our research is still in the publication and still for the ranking, but the society the society need for our impact for our research. This is the valley. So in the death valley of the death, our research is it to move go to the success and the new product. So this is I think we have to collaboration with a lot of the professor as an expert here. This is join the summer course to join together about our research. What kind of the IP we have the research? What kind of department of our department of research? Maybe the professor from the short university will be joined to us to how to develop our research, local resources from Indonesia and local resources from the Seoul. So in here. So this is the STP. What kind of the facility in STP? The academic environment, the research is sample education is started with student. All the invention not only done by the lecture, but the student have the idea. Even though the undergraduate student and the master degree of the PhD, they have the idea. So collaboration with the lecture. That's why the lecturer have to cover our student closely with our student. I think in our department is very closely student in the lecture. Is it right, Bu Sari? Yeah, because we have to discuss, and I have my student here, Dara and the others. We always discuss together with the idea and the others. So this is the research student. It's very important. So who is the join with the summer course? You can collaboration with the student from department, our department, or collaboration with the lecturer in our department. So this is, I think, it's very important. And here we have we have the research prototype patent in here, and here you can have the startup. So look at the right side. After our research, innovative approach become the business. I will show you how the alumni give many business through our research for animal husbandry or animal science. This is the STP. STP is Intermediate Institute to give all the research here. And this is the ecosystem incubation and that. So the STP APB done about that. So we have a lot now. We have done it's about the 500 students. Sorry, startup. One startup collaboration with another scientific, not the same scientific here. So I think this is very important. Thank you, Dara. You already done about that. Animal husbandry. Before I'm going to school, it your at IT because maybe IT Budila doesn't merge. Yeah, it 80. Ya, 80 tahun 80.
Oh,
This is my ya mungkin Budila belum lahir ya.
iya,
Okay,
Bu.
the 80, yeah. We know about that. This only three feed for the feed, feeding, and management. Is it right? Feed, feeding, and management.
Yes.
Now it's not enough. We're not enough for feeding, management, and breeding. So we need the post-harvest. We need the business. So this is the productivity. So the innovative is very important on this because we call the tetragonal. So in here, business, post-harvest, because all our production, all our business in here, it's through our feeding and through the breeding. So I think it is suitable to Ibu Sari and our department. If our department nutrition and feed technology, not only for feed, because we have to know a lot about the animal. So because the breeding, genetic resources, genetic quality, animal breeding, breed and standards. So many Professor Asep Gunawan have the research about how to genetic quality in here. So management, how to farming, breeding and management, reproduction and the others, feeding management. So we call the good farming the practice for the feeding, for the feed here. Feed ingredient, feed quality standard, feed formulation, and feed store is very important. Now we have to call the good manufacturing practice. So this is the final, what kind of our product is suitable, it's best quality or not in our market. So depend on our feeding management, depend on our breeding, and depend for our staff post-harvest. So in IPB, have how to commercialization pathway in innovation in business. IPB always have the innovation launching. So I think that our faculty of animal husbandry or department, you have to give the innovation launching, Ibu Dila. Because it's launching our innovation is branding for us. This is the intellectual property for our lecture, lecture with the collaboration with student. I think not all the lecture can do that 24 hours. So our student have combined in here. So what kind of our conversation here? One, we have to protect IP. Because the IP, intellectual property, this is our like the appreciation for the lecture, appreciation for the student. So if we already protect our IP, nobody one can take our innovation here. So the research have to have protect for IP. If you do them some research in the IPB, the status, legal status, the IP is belong to the IPB. So what kind for the lecture? The lecturer and student is an innovator. And then after that, we have to innovation development. And now IPB have a lot of the like the KDRECA, like PDP and the other innovation development and scale up. Technology evaluation, business incubation, industrialisasi, and community collaboration for social. Nah, so this is, I think the animal husbandry a lot of in the seven in the point end of the seven, because this our paman not only in the industry. In Indonesia, a lot in the community, in the social. So in here, I want to show you, this is commercialization innovation commercialization for IPB. Now IPB have more, I think this is the 2023, and IPB now have a lot of the more than 1,000 for the patent, and patent granted it almost now now is 486 patent granted. So for the retro information now is already 35 in here. Okay. And then I will show you, this is fifth. I saw the innovation for special for feed and animal production. I think, is it it would really happen here? Okay, can I see it? Not yet. Okay, this is the innovation product for our lecture and student combined for our lecture and student. This is the product from Ibu Professor Dewi Apriastuti, BSF. BSF that your division Ibu Dila. And BSF and cat, and for the animal, for the pet animal already get the commercial now. And this is for Ibu Professor Yuli Wafer. Is it Professor Yuli here or the student Professor Yuli? Okay, so this is wafer. This is combined. This is combined wafer for the dairy cattle, and this is Professor Nahrawi. This is the wafer, Professor Nahrawi. This is the innovative protein resources. So this is the hype. This is the breeding for Professor what kind of here? Oh, Professor Asep Gunawan. Domba namanya apa tuh ya, Bella? Gando.
Yes.
Gando. Is it breeding for the Gando, Garut, and Dorber? Yes, that is already have, not only in the field, in the farmer, but in the farm now you have the cane. Thank you, Dara. You give me matada. This is the high protein. Insect based on product Belmont. Jadi want to try this one? I already tried in here formulation. This is the mob feed, and like the mega egg, Professor Astad Busum, Professor Sumiati. I think the formula with the Professor Sumiati, and this is for the inside Professor Dewi. And Professor Dewi, Professor Sri Suharti, Ibu Dila is there now? Ibu Dila, this is your product.
Yes,
Ibu
Prof.
Dila and Kaka, yes, the already collaboration with the Biocycle Indonesia, yes. But unfortunately, I just heard from the Ibu Dewi that Biocycle already closed now. I don't know why. I think it's I think this is the sustainability. So I saw the PT Sugeng Jaya. Sugeng Jaya is our alumni. Is a feed for ruminantia, not just feed for the big cattle and dairy cattle. As we know in Indonesia, a lot of the industry for the chicken and breeding and the others. but maybe only 1 or 2 industry focusing for the ruminantia feed. But our alumni, PT Sugeng is our startup in STP have the ruminantia feed in here. So this is BSF, Catula for Prosultan algae, and this is the omega-3. So Palmofit from Professor Nahrawi, Anuraga, and our team in our division already collaboration with the industry, and they have in the STP now for the feed in here. So maybe this is the more explain about the suitable picture in the before. And this is the prime egg. I think the prime egg, Professor Iman Rahayu, Professor Iman Rahayu. I think the prime egg it become to have the omega-3 or DHA, and the premium egg. Is it based on the formulation? I think our department is very important because the feed and nutrition is support for the production here. So lem rendang, this is the rendang ke Indonesia. This is collaboration Professor Asep Gunawan, and who is from our department, Professor Asep Gunawan and Professor Suharti. I think yes, Sri Suharti. And this is the beef. This is for animal supplement. That's our vocational alumni. And this is the biomap. This is our alumni too. So a lot of our picture here. What kind of the summary here? If we want to sustainability in our livestock or the animal farming in the field, we have to innovative invent not only the research, but the research is suitable to innovation and production. I think it's very important. And then I want to show you, this is the startup. So IPB have the services business incubation model, who is the join the summer course. If we want to be the like the startup, like the businessman or businesswoman, we can join those STP IPB here. We have the model, we have the services to business incubation model in here. And this is business incubation, both workshop and the others. This is our president and president and the president of NUS come to our STP. So this is our alumni. So you can the IPB and the STP will be give facility, the capacity building, mentoring, coaching, business and the others. So we have the building special for our startup. This is in the level 3 or 4, we have the startup special for the animal feed in here. We have the 3 startup here. So our alumni, not only alumni from IPB and alumni from the other universities. So maybe Budila, I think it's very important to bring the member of the startup offline visit to this is special for the startup business according to our field, our scientific in IPB. Oh, this is the startup for all. We have now I think the 500 outward and 31 for inward. And the startup incubation, what of the activity here? And this is the startup room for all. And this is for which one, the animal. Budila, this is our alumni. They have the like we call the cempe, sheep and goat. They have cattle, big cattle and dairy cattle too. They use what is it? Oh, they use the formulation feed from Professor Yuli Wafar. So I saw you. This is the collaboration student, alumni, and our lecture. So I think it's very important how to the scientific become to scale up the innovation and go to the market. I think innovative is very important. So the innovative approach in I think make the animal husbandry grow up there. And this is mentoring for the others, and this is the business, what kind of activity, how to make the how to become the student, a student, undergraduate student, master or PhD program, become to like the entrepreneur here. So this is incubation business process. We bring our startup according to our animal husbandry and agriculture product, bring to the NUS Block 71, make the expo. And then after they have expo in the other, the innovative innovation make the they sign collaboration, like we call the MOU and MOA. This is collaboration with another. So this is international startup expo in Block 71 in Singapore. And they bring like this one, so like for the MOU or MOA. So collaboration not only in national but in international. This is which one? That's our student. Wait, okay, here. I saw the Ibu Dila student here. Which one? Yes. Okay. Oh, it's back. Awang. So the only one. So this is number of startup. So what kind of impact business to incubation startup? The turnover startup is 86%. Employee improved to 40% in here. So IPB have the STP IPB have the facility innovation development. So after the research, we call the invention, go how to innovation development and go to the market. So IPB have the facility for them. This is the facility. We have the teaching industry, and we already have for the guaranteed 3, like this is for food and beverage. And now we have the just building for the halal business incubator building. So you can see all for the startup incubation program here. A lot of the startup. Who has the startup here? Alumni, not a lecture. This is alumni. So for the student, for the all the participants, join the summer course in our department, please. Collaboration with us, because all the startup incubation here, this is the alumni. It's belong to alumni, not belong to the lecture. So the lecture have give the advice. The lecture give the innovation idea, give the innovation invention idea. Collaboration with research. Go to the field. Go to the animal product. Go to the farmers. So go to the market and have the innovation for business. I think animal production, feed animal is treatable for the product of business. So this is maybe only how many, not allowed, but we already have the 500 startup here. So this is the network for the IPB. We networking for the others. Hopefully we can collaboration with the short university. Okay, this is for industry, and award. I think not only we accepted the awards, because the IPB have the three time for the STP, the best in the A accreditation, but we have to improve. We have to responsibility about the award for the IPB. I think this is the Ibu Dila, the IPB innovation market. The IPB we have the how to display all the product to innovation, go to the market. We have the display, not only in display our gallery, but display in the market. We have the digital marketing. I think a lot of the animal production, animal husbandry, and feed already done, and commercialization in the industry. Thank you, Ibu Dila, for give me an opportunity. If there any question for me, I will answer with the happy for all the question. Thank you.
Okay, thank you, Prof. Erika, for giving information about the importance of innovation
Innovation,
to support animal feed and promote the sustainable farming system. Now we will move to the Q&A session. Anyone who wants to ask Professor Erika can use a raise hand button or you can type in the chat box. Anyone?
I think it's direct give the question for me.
Direct question is preferred?
Okay. Okay. Yeah.
Any question? Anyone?
Hi, good afternoon, Prof. Erika.
Yes. Yeah. This is Emily.
Yeah, this is
Hi,
Emily
I'm
from the
Emily.
Philippines.
Oh,
Yeah,
Yeah,
I know you.
it's
How
nice
are
to
you?
see you again.
But I cannot see your face here.
Yeah. I'm sorry. I actually I just stopped by just to ask this question because I'm on travel. So
Okay.
it's
Okay.
very nice
It's okay.
to see you and hear your presentation.
Yep.
I just would like to know, how does
Yep.
IPB
Yep.
particularly the Faculty of Animal Science maintain
Yep.
its innovation process, like
Mhm.
from experimental studies to intellectual
Mhm.
property protection? Because I see
Mhm.
there's a lot already of
Go to the market.
commercialized product. Yeah.
Yeah.
Could you share a brief overview of the steps that have contributed to your success in innovation? And I'd like, I asked
Mhm.
this kind of question because I'd like to inspire and encourage my students who are here with me in this Zoom session. So with me are my 6 students. And
Yep.
yeah, I am really very proud that once I have visited and we have collaborated with IPB. So yeah, just a short or a brief view or overview of the process. Bu, thank you so
Yeah.
much.
Okay. Thank you, Emily. When you come back again to our division?
I do not know yet, but I hope I can bring my students there so that they can also
Hopefully.
see what IPB can offer.
Okay. Okay. So for the because we have to one perception. After the research, this is not innovation. The result of research this is the we call the invention. Invention, is it right? I remember when we have the meeting for the IPO, the Intellectual Property Organization in Cebu, Manila. We consider that invention different with innovation. Invention is result for the research, not yet to innovation. What kind of innovation is it? The innovation is invention plus protection IP. So that's why IPB have IPB have the facility how to protect IP. As we know, if we do some research in our university, all the invention and legal status is belong to university. So what we have the function here. So the innovator, like the lecture student, this is the innovator, not the belong to the status the IP. So we have to protect IP. What kind of process? IPB have the Science Technopark, and I explained before. Science Technopark, or we call the Institute of Science and Technology, is one how to register the IP. Register the RP, we call the Directorate General IP, DGIP. DGIP protect our registration. If we already have the registration, nobody can take our innovation. This is the protect Ibu Mrs. Amy. So the protect IP, this is the process. Not one by one lecture go to the DJKI, but the institute or the ISTP, according to me, protect the IP registration. After the registration, we have the script, the patent granted. And then after the patent granted, we already have the certificate. So the certificate, the right of the certificate, this is the IPB protection certificate. We call the certificate patent granted. It is the process. And then what kind after we have the certificate, we will be develop the innovation. I think not all the invention is become to have the product. They have become to production because many of the one patent, one or two patent is not yet become a product. So we have to develop again about our invention. So we call the scale up innovation development. And after that, if the already have we call, as you do you know about the TRL, Ibu Emi? TRL, this is level for the marketing. So this after we have the TRL 7 until the 9 is go to the market. So okay, we have the collaboration with the industry or society, but no one by one in the lecture because IPB have the Institute of Science and of Art. Is it right? Is it I answer your question?
Yes, thank you so
Yeah.
much, Bu. Yeah, it
Yeah.
enlightens us. Yeah, but in a way, your process is still, I think it's effective, and yeah, you're successful in having those kind of innovations already protected. Thank you so
Yeah.
much. Hope to see you
You are
soon. And
welcome. Yeah.
yeah,
Thank you.
regards to
Yeah.
all the faculty in the Paternacan facultas. Thank you, Bu.
Okay. Thank you. You are welcome. Yep.
Okay, now we have other participant who want to ask Liana.
Where is the
You
Liana?
can ask directly.
Okay.
Okay, good afternoon, ma'am.
Good afternoon. Yeah.
Before that, let me introduce myself. My name is Liana Wahyuni. I'm from Agriculture and Livestock Department, Musirawas Regency Government.
Musirawas, is it the
yes.
South Sumatra? Oh, this is the same with me. Yeah.
Yeah.
I'm from Palembang.
Oh, I'm from
Okay.
Lubulingao.
Oh, Lubulingao. Okay. What kind of your question? Yeah.
Okay. First, I'm so interested in your presentation, and you be my inspired. So in our region, so many farmers use soil palm oil byproduct.
Palm oil, yeah.
Yes, as livestock feed in addition to green forage. However, as we know that solid palm oil residue can disrupt livestock metabolism if not processed properly.
Yeah.
So from my further research, I found that some people process solid palm oil residue into products like Sakura block.
Yeah.
However, this increases production costs, which reduces farmer interest in using such processed feed. So what is the best and most practical solution to address this issue, improving the nutritional value and safety of solid palm oil residue as livestock feed while keeping production costs low and ensuring farmer acceptance? Thank you.
Okay, you are welcome. You mean this is for the palm kernel oil? Yeah.
Yes.
Yeah. I think the IPB already done about the palm kernel. Palm kernel, because for the palm kernel, and the other for the industrial, the industries, we do not say they are the waste, but byproduct. A lot of the byproduct, bipolar palm kernel, because you see that the palmofit from Professor Nahrawi, we call the from the bees. Bees is the cell content for palm kernel. All we call in Indonesia bungkil inti sawit. Okay, is right? Bees.
Yes.
So we use the not only sluts. You said the slut. Yeah, I have the formula for combination cacao and palm kernel. So I use our formula when I get the PhD program in Sumatra, and in Sumatra in Riau. We know in Riau SIKA. SIKA,
Yes.
This is a system integrasi kelapa sawit. I have a lot of 7 2,000 beef cattle there. All from the we call the Kuantan, Kabupaten Kuantan, that's Cikadel. They use the palm kernel byproduct. They use the bees, and they use we call the pelepah, and we use the slat. Okay, so we use they have the three kind of the feedstock from the palm kernel, and combined with cacao pod. Cacao, you know that cacao pod? This cacao pod it is more than 60 I think more than 70%. This is the byproduct. But cacao is very dangerous. If you stay in the cacao tree, they have malin kundang. I call the malin kundang. Because the cacao tree, the cacao pod will be destroyed the strong steam and we call leaf for the cacao. So that's why cacao pod you have to go anyone from our gardening in the cacao. So this is the combine. I have the formula. When I use the combine from the cacao byproduct and palm kernel byproduct, I have the ADG, average daily gain, and for the beef cattle is 1.5 per kilo each day. I think it's very important. So then you ask me for the palm kernel for the slat. This is the slat. You mean the slat? I think slat. Yes, the slat. We call it in Indonesia lumpur.
Yeah.
Yeah, this is lumpur. Liquor lumpur or slat is very dangerous because they didn't filtration about the oil. They didn't filtration about the water in there. So when we want to use, you have to we have to filter for the slat and for the oil there. But I think they're not in zero. That's why the slat not more than, I think it's only 5 until 20% you can use for our stuff, because it's very, I think a lot of the fiber and a lot of the oil, because the unsaturated oil in there for the slat. Okay, is it right?
Okay.
So I think an IPB not only for feed. If you want to see it in our gallery, I have the clothes, I have a hat, I have the shoes. We have a car with the rompi tahan peluru, and we have the what is it? Rompi tahan peluru. So, and we have the hijab from the byproduct palm kernel. So a lot of research in IPB about the palm kernel. Okay, Juliana.
Okay, thank you so much.
Yeah, you are welcome. Yeah.
Okay, I think we have
I saw from
I saw one
the
person,
others.
one,
Yeah.
one.
Yeah.
Oh, Dr. Moses?
Which one? Dr. Moses or the
Yes, hello, Professor. Thank you for this opportunity.
Yeah.
Yeah.
please.
Okay. Yeah.
Yeah,
Moses. Hi.
yeah. Hey, I'm Wambale Moses, Uganda.
Uganda. Okay.
Yeah, and my question is, are those nutritious supplements you've shown us, are they on market? More especially if they are on market, have they reached mostly East Africa or Uganda, so that we can acquire them and teach our farmers on how to use them to supplement
Yeah.
to their feeds and give them to animals, so that we can improve on nutrition to animals in Uganda.
I think it is the same from Indonesia. In Indonesia, we have it is similar with Indonesia. If the company selling for the feed supplement on vaccine on the other for the animal, they have to have like the technical assistant how to use the supplement, because the doses in the supplement like feed additive and the other is very important. because not the bigger dose for our animal. That just small animal, the small doses is very important for our animals. So that's why feed additive, feed supplement. The company have produced the feed additive and the feed supplement should have the technical assistance, how to use it and how to remove and how to give the withdrawal time if you have the how to protect the healthy in there. So the direction, the feed supplement and the direction in feed additive, you have to give in the bottle or the other packaging there. Because this is the regulation in Indonesia, feed additive and feed supplement. For the feed or healthy food and for the food, we have the three kind of the regulation in Indonesia. One, they have to comply the Indonesian national standard. All the nutrient, all the doses in the we call the SNE. They have to comply there. And then second, they have to comply the regulation how to make the healthy for the animal. We call it like the certified for like the agriculture chemistry, because this is for the animal. For the food, we have to certify, we call the BPOM. This is for the healthy. And the third, I think maybe is it the similar with Uganda? From the like the we call the halal toiban. That's halal. It's very important too for the feed supplement and the feed additive for animal, even though not direct impact, but indirect impact, like the many vaccine and many feed additive. This is also suitable for our regulation and our religion in Indonesia. I think they're similar with Uganda. So the three kind of the regulation there. Okay. That's why the alumni from the IPB, the alumni for our department, the first they have to fill for the employee as the technical assistant in the company, because the TR or technical assistant in the company, they have to have knowledge about the healthy, about feed, about animal and the other. So I think for our student learning a lot about the knowledge in animal husbandry. It won't go to the market. Thank you.
Okay.
Yeah.
I think the answer is enough. Anyone?
thank-
Maybe one more question? We still have 5 minutes. Anyone?
I saw Ibu Welas here.
Ibu
They make
Welas.
the Welas. They give me note. Hi, Bu
Oh,
Welas.
yes.
Yeah. But I forget the face for Ibu Welas. Ibu Welas, where are you? You send me message. Hi, mom.
She is
Okay.
committee also. The
Yeah. They
committee.
give me. Yeah. Oh. Okay.
Okay.
Yeah.
I think no more question from the participants.
Thank you.
We can end the Q&A session. Thank you, Prof. Erika, for the engaging engaging session and the insightful answer, and also for all the participants during this session.
Okay.
Okay, thank you so much, Prof. And before I end this lecture session and bring back to the MC, I would like to express thank you for
You
all.
are welcome. You are
Yeah.
welcome. So thank you, Ibu Dila, Ibu Sari, and Ibu Lilis already invite me. Even the one, thank you to my daughter Dara. Always help me because a lot of the duty for me, a lot of the responsibility for me. But Dara already ready for your mom. Make all running well. Thank you, Ibu Sari. Ibu Sari, I still in the Taman Kancana until in the evening because I have the meeting here. Thank you very much for all. Enjoy the summer course. Also invite for the excursion. If you want to visit the site Park, so send the letter because the other department already sent the letter to see the site Park in IPB University. So the excursion will be, the participant will be, so the alumni, our alumni pro animal husbandry have the company. It's already have the company. They have the big salary more than the lecture there.
Yeah.
Okay, thank you very much. Okay,
Thank
bye-bye.
you so much,
Nice
Prof.
to meet you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. And I also want to thanks for all the participants for the active participation. And I conclude this session. See you again in next session. I will bring back the floor back to the MC Dara. Thank you.
Thank you so much, Dr. Dila, for the discussion. Also, thank you, Professor Yun and Professor Erika, for your engaging and insightful presentations. Before we end, I think we will have a group photo again. For the participants, please, I kindly request to enable your camera and share your best smile. Okay, for the first slide, 3, 2, 1, cheese. Next slide. 3, 2, 1, cheese. Again, thank you so much, Prof. Erika and Dr. Dila, for today's session.
Bye-bye.
Bye-bye.
See you.
Okay, this concludes our day 2. Don't forget to fill out the registration form and complete tasks related to today's topic, which is available through the OCW. Our next course will be held on Monday, July 14th. So happy weekend, everyone, and see you next week. Assalamualaikum warahmatullahi wabarakatuh. Thanks.